It is really a truly pleasure to being able to be part of this collaboration. Thanks, Brian and Adam, for the introduction. And it's really my great honor to be able to represent the whole CBTN and the CPTech HOPE project working group to share some of our latest findings here. So I will first give some background about the CPTech consortium and its collaboration with CBTN, and then how that motivates this HOPE study on high-grade glioma in children and young adults. And then we'll show some preliminary finding. Just as Brian pointed out, it's a very ongoing effort. Okay, so I figure probably everyone knows TCGA, which is an NCI-initiated consortium using deep genomic profiling strategies to study cancer. And the CPTech is a sister consortium to TCGA, which is kind of established a little bit later than TCGA, but the focus is adding the protein and the PTM to the puzzle such that we can better understand all those diseases. So in CPTech, we are using this deep integrated proteomics genomics profiling strategies to study tumors that we use in maybe almost 10-ish platforms, including those for genomics, and as well as the proteomics platform to profile global proteins, phosphoproteins, and many other PTMs. And we combine the information in the tumor versus the adjacent normal to trying to see whether we can understand those diseases better. I think in the past 5 years, the researchers from CPTech has been really successfully demonstrated that by adding the proteomics and the PTM into this puzzle, we indeed gain new insights of all the diseases. And those work has been recently kind of reported by a series of papers of this 10 tumor type in cell or cancer cell. And specifically, I want to point out this one paper on pediatric brain tumor. So this is the project that Brian just mentioned. It's stemmed from this beautiful collaboration between CPTech and the CBTN. So in this project, we utilized this integrated deep proteomics genomics profiling strategies to study 7 major histological types of pediatric brain cancer. So there are many exciting findings from this project, but I want to, emphasize one perspective. Actually, that is from Adam initially. So the idea is that while we all know there are a lot of between patient tumor heterogeneities, like each individual tumor seems to have different mutations, different copy number changes, but those combinatory complexity at the DNA and RNA level may ultimately represent a common proteomics solution at the pathway activity and the signaling level. And this perspective kind of is beautifully validated in this cross-disease analysis of pediatric brain tumor, that we, using the proteomics clustering, we are able to reveal actionable biological features spanning different histological boundaries. And also by using protein and phospho PTM data, we are able to characterize some kinase activities, which also brought us new insights into druggable targets. So I cannot emphasize enough how wonderful our teamwork is that we have in this project. Really grateful for the leadership from Adam, Brian, Philip, and other from the CBTN, as well as the NCI CPTech leadership that make this project feasible. So we enjoyed a lot, and we are very happy that this proteomics genomics strategy seems to be successful when we use it to studying the pediatrics tumors, and we definitely want to keep the momentum. Okay, so in the pediatrics project, there is one histology type, which is the high-grade astrocytoma or high-grade glioma, HGG. So this is a histology type that is of particular interest because it is the most common malignant brain tumor in children. And the survival outcome is the worst among the 7 types. And by now, there's not many effective altering therapies that are available for the kids based on genomics features. So we really are hoping to maybe identify some new therapy possibilities for those children based on maybe the functional biology of the tumor. But in the original pediatric study, we only have 26 HGG samples. So we definitely want to expand the cohort such that we can have more thorough investigation of this tumor type. Another important aspect is how this HGG tumor at different age actually has very different characteristics. Thanks, Brian, to actually created this table, which nicely characterized how the HGG at the childhood, at the young adult age range, and at the adulthood, which is often GBM, differ from each other. Especially, it's kind of well agreed that the adult GBM and the pediatrics HGG has many different mutation driving events. But the understanding of the AYA patients actually is still very limited. And it is very interesting to understand whether the HGG at different age span really share the same or different biology. So that is kind of motivate us to initiate this HOPE project. And really thanks Adam and Brian and other colleagues from CBTN that to have another great opportunity to collaborating together. So another good aspect is that by leveraging the information and the data from the pediatric study, as well as the adult GBM study from CPTech, we kind of have a unique opportunity to comprehensively characterize the interplay between the tumor process as well as this brain developmental process. Okay, so for the HOPE cohort, we received the frozen tumor tissue samples from 87 pediatrics and AYA HGG patients from CBTN and UPenn. And those pie chart kind of illustrated the composition of those patients and samples. Specifically, there are 61 samples is coming from patients in the pediatrics age range, while 34 samples is coming from AYA patients. This figure further illustrated the age distribution in the HOPE cohort, in the pediatrics cohort, and in the GBM cohort. So by leveraging data from all the three studies, we kind of have better sample size to really understand this HGG in the AYA age range, which is kind of a rare disease. So thanks Matthias from CHOP and Nicole and Dimitri who helped with the survival analysis of this combined cohort of three study. As illustrated by this KM curve, when we stratified patients into this pediatrics and AYA and adult age span, we can see the AYA age group actually has the best survival, and the differences between AYA to pediatrics and adult is significant. So this is quite intriguing, right? So the fact that the HGG patient of different age group had different survival outcome supports the hypothesis that those HGG may utilize different biological mechanism. So therefore, in this HOPE project, we hope to utilize deep proteomics and the genomics profiling technology to pull back the curtain of the interplay between the tumor process and the developmental process of the brain. So this is truly a kind of team effort that between CPTech and the CBTN, we planned to perform deep profiling, multi-omics profiling using 10 different genomics or proteomics platform, including the global proteomics, phosphoproteomics, and RNA, microRNA, single cell RNA, whole genome sequencing, methylation, as well as some directed proteomics platforms to validate some of the markers or pathways that we found to be interesting or important from the project. So while many data profiling is still ongoing, we did finish the protein and phospho profiling very lately. So today I will share some interesting findings from this blog. I think Dr. Aaron Dice will also share some interesting finding from the single cell in the next presentation. Okay. So to generate the global and phosphoproteomics data, multiple teams from CBTech has contributed, including the lab from Fred Hutch, the lab from PNL, the lab from Michigan, and also the Sinai team. In the end of the day, we obtained the global proteomics and phosphoproteomics data for almost 9,000 proteins and 20K phosphosites. So the first thing is we try to look at the landscape of the global proteomics data, which is shown in this heat map. So each column is a sample and each row is a protein. So interestingly, we see there are 3 distinct kind of groups that is suggested by the protein data that it seems to have a slightly different kind of protein activity characteristics. And this box plot illustrated the age distribution of this 3 proteomics cluster. And as we can see, this first cluster seems to be older than the second and the third cluster, which is more enriched of pediatrics kids, patients. But if we just focus on the AYA group, which are the patients with age greater than 15, I think 50% is from this cluster 3, and 25% from class 2, and 25% from cluster 1. So there are also some other clinical characteristics differ from these 3 groups of tumors, including the tumor location. We have more midline in the 3rd group, and also the tumor resection seems we have more total resection tumors in the 1st group, and the tumor multiple tumor location, which is basically here. So some other thing I want to mention is we also did this analysis using the phosphoproteomics data. Actually, we observed very similar 3 clusters, and we also performed this exercise using the combined HOPE, GBM, and pediatrics samples, and we again see very consistent kind of 3 cluster structure. We then try to characterize what are the pathway activities of the 3 protein clusters, and we can definitely see there are some very distinct pathways like super upregulated oxidative phosphorylation, and the synapse plasticity is super upregulated in the 1st cluster. The cell cycle seems to be more upregulated in the 3rd cluster, while some antigen and complementary pathways is more upregulated in the 2nd cluster. So that is interesting. There seems to be indeed 3 groups of different tumor. Then the next question is, are those differences really clinically meaningful? So the first thing we look into is survival. So thanks for the CHOP team and also the other data analysis team members, Nicole. We performed extensive survival investigation. Especially, we try to stratify the survival investigation into different age range, as we show on this figure. So I will just point out the most striking part. So first of all, within this AYA age span, we definitely see the 3 proteomics group actually has very different survival outcome. And it seems this order is not reflected in the other age range, which kind of suggesting, yes, this developmental stage, like different developmental status of the brain, probably has a big impact on the tumor, and maybe the tumor is using totally different mechanism to develop at different age span. So this is really motivated us to look deeper about what's the role of this developmental stage in those tumors. And this is really complicated, I would say, for studying the pediatrics and young adult brain tumor, because if we just do a very simple investigation of the data, we can see more than half of the proteins actually are greatly associated with the age, which is reflecting the different developmental stage. Therefore, when we see those differences, different structures in the protein data, it seems to be a little bit difficult to really tease out what are the molecular characteristics that is reflecting the age, or what are the molecular characteristics is reflecting the tumor process or the interplay between the tumor and the age. So that is actually quite a bit challenge. But whenever there is a challenge, there could be an opportunity. So in brain tumor study, actually, one difficulty is that it's not very easy to obtain normal or adjacent brain tissue. So this actually is a big obstacle in our previous brain tumor project. And in addition, even when there are normal brain tissues, because the tumor brain tissue and normal brain tissue are often consist of different cell type, then the protein abundance usually is not directly comparable between the tumor and the normal tissues. However, here, what is interesting is that because we are studying those brain tumors across a spectrum of age groups, and then the strong dependence of the protein and the PTM activities on the age actually provide a nice opportunity to screen for tumor-specific signals. So I will elaborate what that means. So in this figure, we are taking another data set, actually it's a proteomics data set from another research group from Mount Sinai, conducted earlier, based on normal human cortical tissues. And that data set is also studying a sample that crossing a large age span. And using that data set, we are able to kind of characterize different groups of proteins that seems to have different trajectory changing along age. So in the normal tissue, we see there are clearly three clusters of pathways that some is basically very high in the infants or young baby age and then decrease. And another group actually is increasing in the first 10 years and then decreasing. And the third group is continue increasing and then start to decrease after age 40. But when we look at the similar kind of parallel way of the data from the tumor samples from the HOPE project, actually we see very different trajectory. We don't see this increasing and decreasing kind of group of pathways. And some of the member in that group is synapse organization, transmission across synapses, oxidative phosphorylation, and the regulation of neurotransmitter levels. So this is just a limited number of examples, but the point is that indeed there are biological processes that in the tumor is having a different trajectory than that of the normal. So this could be an evidence to say, okay, yeah, maybe that pathway indeed is due to tumor activity. So this is a more detailed example that I'm focusing on this one categorical category regarding synapse. And I'm showing multiple curves. This gray curve is coming from the normal sample of that reference data set. And then the three color curve is corresponding to the three protein clusters from the HOPE study. So we can clearly see that the purple and the blue, which is the protein cluster 2 and 3, definitely has a different shape compared to the normal brain tissue, which is suggesting, yes, most likely the synapse-related pathways is playing a different role in the tumor than in the normal brain tissue. And this actually echoes the latest literature findings about how the glioma cells can hijack the neural cells to develop some neural glioma synapses to support the tumor development. So this is a very powerful way to using the change along age, like this developmental trajectory, to try to identify tumor-specific signals. And this is just two other examples that illustrating two other pathways seems to be particularly abnormal in the tumor 3 cluster. So that is actually the cluster with the worst survival outcome. So we are currently still in the middle of investigating those pathways. And of course, I want to point out that this is just a very ongoing investigation, because we still need to have all the genomics data to understand what are the H3 mutations, IDH mutations, and other important factors to really figure out what's going on in those tumors. Okay, so that is just a brief trailer of the current HOPE project. And we are very looking forward to the next step of large-scale integrative omics learning, that once we got all the data from all the platforms in place, we will perform many other integrative investigation, trying to really figure out, okay, how this brain development and the tumor process are interplay, and how we can better help those HGG patients. In the end, I want to again emphasize those exciting projects will not be possible without the great teamwork and the leadership from the CBTN as well as CBTAC. So it's really a joint effort from many teams, and also thank the patients and their families for contributing the sample to make this possible. Okay, I will stop here and happy to take questions.
Thank you very much, Pei. That was a really wonderful insight into sort of the way that these projects evolve, and we're excited to see where it's headed. There'll be a lot more data coming. We're running a bit behind. So.
I know we're running behind, but I just want to highlight one point, just to connect to, some of the, one of the exciting things that Dr. Weinberg highlighted for those of you who were listening at the time was that the person who figures out differences between pediatric and adult mechanisms will win the Nobel Prize at some point in time. So perhaps Pei is fortunate of doing that. And likewise, I suspect Erin. And you'll notice that there's Pei used the term Project HOPE in her narrative. And I just wanted to highlight the connectivity between this. Oftentimes in CBTN, we're leveraging multiple streams of NCI funding and support to bring things together. And so the work with Pei and CBTAC is actually using the same specimens that overlap with, I think, you'll hear about next from Erin, just to connect some of the terminologies.